MCAT 2017 Cram, Biochemical and Biological Foundations of Living Systems. Passage 5, Preventing Lipopolysaccharide Formation in Cystic Fibrosis Patients. As you view the reading of the passage, you'll notice some highlighted snippets of text. What I want you to do is garner meaning from these specific selections in order to answer the questions that follow. Good luck and happy reading. Paragraph 1. Young patients diagnosed with cystic fibrosis are at high risk of developing debilitating pulmonary infections caused by the bacteria Pseudomonas aeruginosa. In these patients, P. aeruginosa secretes a mucus substance called alginate and overproduces a virulent lipopolysaccharide, both of which contribute to chronic pulmonary infections. The basic chemical structure of an LPS molecule is drawn in Figure 1. Figure 1, the basic chemical structure of an LPS. We have one subunit, phosphates. This looks like a disaccharide bond. Fatty acid chains, okay. Paragraph 2. A precursor of LPS is glucose 1-phosphate, which can be created when the enzyme phosphoglucomutase, PGM, rearranges glucose 6-phosphate. A precursor of alginate is mannose 1-phosphate, which can be created when the enzyme phosphomonomutase, PMM, rearranges mannose 6-phosphate. In P. aeruginosa, both the conversion of glucose and mannose are performed by a single bifunctional enzyme identified as PGM-PMM. Since both alginate and LPS cause the infection, researchers are actively looking for mechanisms to block the PGM-PMM enzyme and thus stop their production. Paragraph 3. The blocking of PGM-PMM has other benefits too. For example, when cells break down glycogen, the major product is glucose 1-phosphate. Large supplies of glucose 1-phosphate can be converted by PGM to glucose 6-phosphate, which is a beginning molecule for both the glycolysis and pentose phosphate pathways. If the PGM enzyme is blocked, glucose 1-phosphate will not be participate in these metabolic pathways, and the cells will not generate the critical resources needed for survival. Experiment 1. The enzyme kinetics of PGM-PMM were investigated, and the results are detailed in Table 1. So here we see the Km, the Michaelis-Menten constant, and the Vmax. So Vmax is reached quicker on the, PMM aspect of the enzyme versus the PGM aspect. It takes a higher substrate concentration to reach Vmax on the PGM side and a lower to reach on the PMM side. Okay, all right. Experiment 2. Through a series of column gel filtrations, the PGM-PMM molecule was extracted and then processed through SDS page. Figure 2 shows the SDS page gel of the process enzyme. Lane 1 is a standard protein ladder. Okay. Lane 2 is PGM-PMM prior to filtration, And lanes 3 through 5 are progressive purifications of the enzyme. Lane 5 is the final purification of PGM, PMM. All right. If electrophoresis was performed without SDS, what would be a reasonable location for the enzyme PGM, PMM? Is it near letter A in figure 2? Is it near letter D in figure 2? Is it near letter C in figure 2? Or is it near letter B in figure 2? You decide. I'll give you a moment to think. All right. Okay, so SDS, which is short for sodium dodecyl sulfate, inserts itself into amino acid chain and has basically a net negative charge in buffer solution. SDS also interrupts the the tertiary structure of a protein, making a straighter chain of the polypeptide. Okay, so here you see the net negative charge is what causes the force of attraction between the anode terminal somewhere down here of the electrophoresis machine and the negative charge on the polypeptide. Okay. Without a net negative charge and a straight shape, not just a negative charge, the amino acid chain, it won't migrate from here to there. So it won't migrate quickly as well, okay, through this gel solvent. So the molecule will appear significantly larger than it actually is. Therefore, the most reasonable answer would be near letter A in figure 2, because if this, is 45 kilodaltons this far, then this is traveling a little bit slower because it's larger. It's like, a fat person trying to outrun a skinny person. Normally, you would expect the thin person to win. You could compare it to that, I guess, so that it makes sense. It kind of seems antithetical that the smaller thing gets further along, but that's just the way it is, okay? All right. So PGM, that's phosphoglucomutase, converts glucose 1-phosphate into glucose 6-phosphate. If glucose 6-phosphate molecules enter the pentose phosphate pathway, which one of the following is produced? Is it going to be A, NAD+, B, NADH, C, NADP+, or D, NADPH? I'll give you a moment to think. NAD, nicotine adenine dinucleotide, and NADH are most prominently used and generated in glycolysis, the citric acid cycle, and the electron transport chain. These molecules, they don't participate in the pentose phosphate pathway, okay? The pentose phosphate pathway generates reducing molecules that can be used in reductive biosynthesis of other molecules. To be a reducing molecule, there must be electrons that can be transferred to the molecule, okay? Thus, the correct answer choice is NADPH with its extra hydrogen, or hydride rather. See here, there's a transfer of electrons. It's the reducing molecule produced in the pentose phosphate pathway as pictured here. Among other things, this pathway also produces the ribose needed for synthesis of nucleotides, okay? All right. Where in the cell would LPS lipopolysaccharides likely be found? Is it A, inside vesicles ready for exocytosis? Is it B, attached to the plasma membrane of a cell? Is it C, attached to the nucleolus? Or is it D, free-floating in the cytoplasmic fluid of a cell? I'll give you a moment to think. This requires standalone knowledge outside of the passage. Okay, so LPS, lipopolysaccharides, have long hydrocarbon tails, the fatty acid tails here, that will be hydrophobic. Okay, they won't mix with water. So the hydrocarbon tail of LPS will not exist in the polar environment of the cytoplasm. Okay, LPS also has polar groups, pictured here in red. Okay, so the polar part of the molecule will be able to exist in a polar environment. Also, the phosphates are polar in green, and these two, what appear to be glucoses, are polar as well. Okay. All right, so yeah, they're going to exist in polar environments, just not the hydrocarbon tails. So basically, LPS lipopolysaccharides are located on the exterior of the plasma membrane. They're depicted as these little squigglies right here. The structure and shape of LPS is similar to a phospholipid in that there's a polar region and the nonpolar region. The tails in black are the nonpolar region, okay? The fatty acid tails, since they're nonpolar, can insert themselves into the phospholipid bilayer, while the polar glycosylated regions are located outside of the cell. Okay? All right, so that's the correct answer. Answer choice B, attached to the plasma membrane. All right. Following the data in Table 1, does this graph accurately depict the velocity versus substrate concentration graph for the enzymatic kinetics of the enzyme PGM-PMM, phosphoglucomutase, phosphomanoamutase? Is it A? No, the graph implies that the enzyme has equal affinity for mannose and glucose, and the Vmax for each substrate is not correctly drawn. Is it B? Yes, the graph correctly shows the enzyme has equal affinity for glucose and mannose, and the Vmax of each substrate is correctly drawn. Is it C? No, although the graph does correctly show the enzyme has a greater affinity for mannose than glucose, the Vmax for each substrate is not correctly drawn. I'll give you a moment to think and refer back to Table 1 in the passage. All right. Okay. Okay, so for those of you who aren't familiar with enzyme kinetics, or reaction kinetics, this could take a minute. So let me break this down for everyone, okay? Km is a measurement of how quickly a molecule dissociates from the enzyme. In this case, the enzyme is PGM, dual function enzyme. The higher the value, the more quickly dissociation occurs. But note, many textbooks consider Km in the opposite way, okay, where Km describes the enzyme's affinity or attraction for a molecule. In this reading, however, Km means a low affinity, and a low Km value means a high enzyme affinity, okay? Affinity being the inverse of dissociation. So just take that in for a moment if you need to. Via the classic mathematical derivation of the Michaelis-Menten equation, this is the Michaelis-Menten constant, where the sub m stands for Michaelis or Menten, I'm not sure, but you get the point. It works out that substrate concentration is equivalent to Km, okay? So, and it also works out that V initial is equal to 1/2 V max, all right? So looking at the substrate concentration value that corresponds to the 1/2 Vmax value for both substrates. This graph has the same value for both mannose and glucose. When I say value, I mean the substrate concentration. It occurs around like 50 micromolar, okay? Yet, according to table 1, the values of the substrate concentration should be different. It should be either like 15 or 22, which is somewhere over here, okay? So yeah. All right. The Vmax values for each substrate located in table 1 should correspond to horizontal asymptotes on the kinetics graph, namely 28 for mannose and 60 for glucose. We can see that the glucose 1-phosphate portion of the enzyme is way lower than 60, and the mannose 1-phosphate portion that processes the portion of the enzyme that processes mannose 1-phosphate is way higher than 28, okay? So this is wrong. This is not an accurate kinetics graph of the enzyme PGM, which is phosphoglucomutase and phosphomonomutase for short. The graph implies that the enzyme has equal affinities, so equal Km values for the mannose and glucose, okay? Moreover, the Vmax for each substrate is not correctly drawn. One is overdrawn and one is underdrawn. A correct graph will have the glucose 1-phosphate curve above the mannose 1-phosphate curve, okay? Because the Vmax for glucose is higher. So basically the correct answer choice is going to be answer choice A. What else can we critique about this drawing? And moreover, each substrate has a different Km value, so each curve should have a different substrate concentration that corresponds to the 1/2 V max value. I should specify that the correct Km occurs at the 1/2 Vmax value. Okay, I don't know if I specified that. For those of you who aren't already familiar, you should be familiar. So the graph both doesn't have the correct Km values, and nor does it have the correct Vmax values, okay? The graph in this question suggests, again, that the enzyme has equal affinities, or Kms, for both glucose and mannose, which is not true based on Table 1. And this graph also suggests that mannose has a higher maximum velocity than glucose, okay? All right, so this is wrong. Using the results from the SDS page experiment presented here in Figure 2, what is the approximate size in kilodaltons, kDa for short, of the enzyme PGMP that is found in Pseudomonas aeruginosa? Is it A, 30 kilodaltons, B, 15 kilodaltons, C, 300 kilodaltons, or D, 50 kilodaltons? I'll give you a moment to think. All right. Okay, so PAGE stands for polyacrylamide gel electrophoresis. And in this process, there are small pores that allow the molecules to travel through. It's basically more challenging for large molecules to travel through, or basically transverse the small pores. Longer fragments will travel through at slower rates, and the length of the polypeptides is proportional to the molecular mass, which is measured in kilodaltons, okay? So we use the molecular mass of the molecule as a proxy or substitute for the length. So the distance traveled by most polypeptides, again, is linearly proportional to the logarithm of their mass. So the most reasonable answer in this case is going to be 50 kilodaltons, and it is actually the real measurement made by the research team in this original experiment. The fragment has to be larger than 45 kilodaltons noted here, because here goes the final purification, first of all. And the fragment traveled less of a distance. This means it's heavier. Okay, so if it's heavier than this one that traveled further, it has to have a higher mass or a higher value in kilodaltons. Okay. However, the fragment will not be an order of magnitude different in size, because again, the relationship between distance traveled and length of the fragment is linear. So it's not going to be like greatly bigger, but it's still a little bit heavier. Okay. So that's why the answer is not 300 kilodaltons versus 50 kilodaltons. All right, that's it. You made it.